Hello, welcome back to Holistic Headspace. I'm Dr. Keeley, licensed naturopathic doctor.
And I am Morgan, naturopathic medical student.
We're going to be talking all about the microbiome. So last time we left off talking about the gut-brain connection and did a little bit of an introduction to the microbiome and how it's working with our nervous system. Today, we're going to talk about kind of the foundations of establishing the microbiome and maintaining good microbiome health, and just more of a broad overview of everything you kind of need to know about the microbiome and what we know, what we don't know, that sort of thing. And then we'll go on a bit deeper dive in some future episodes on specific things like that. So, Morgan, you want to kick us off? What is the microbiome?
Spoiler alert, there's a lot. Yeah. So spoiler, there's a lot of things that we don't know, right? First of all, this is a huge developing area in medicine and science right now. So just to start off that way. But really, so what is the microbiome? So it is the community of literally trillions of microorganisms. So this could include bacteria, this could include viruses, fungi,
Archaea.
Sorry, I forgot right when I got to it. And protozoa, right? And these are all living in and on the human body. So, and we're talking mouth, throat, stomach, of course, the GI system, but we're also talking about skin. We're talking about lungs. We've seen that the brain might have its own microbiome. Of course, like in utero, when we're growing a human baby, though, there's its own microbiome in there. So there's all these unique kind of environments all over the body that have different levels of these microorganisms. And they all play a role in pretty much everything we do, right? So they play a role in digestion, in our immunity, they play a role in our mood, our metabolism, our health, and of course, what we love to talk about here, nervous system regulation, right? So most of the microbes that we really have studied and really understand are located in the gut. And just in general, most of them are located in the gut, particularly in the colon, but they're everywhere. So when we talk about the microbiome, we are referring to really the collective genomes. So when we talk about genomes, we talk about genes, genetics, DNA, right, of these organisms and their interactions with everything. So this is a super complex system that we're still really learning a lot about. Why is this important, right? Because it literally influences everything. And the more we learn about them, the more we see how much they actually are influencing us as a human body. And we see that changes in this microbiome are linked to diseases including obesity, diabetes, autoimmune disorders, mental health disorders, and even as much as cancer. So lots to look at here. Maybe Keeley, you can talk to us a little bit about kind of what's the history of the microbiome in research? Like, how did we get on this trajectory?
Yeah, honestly, we have so much data right now in mouse models. So tons and tons of literature on mice, and they've actually grown, I forget what the term is for the mice that don't have any microbes.
Germ-free.
So they've grown germ-free mice and then given them microbes to see what happens with specific bacteria. And these mice that didn't have any microbiome, they suffered so much that they actually had to stop doing that and only work with mice that they could use antibiotics to knock out most of the microbiome or the gut microbiome. I guess you should specify because there's microbiomes in all different organ systems, but the gut microbiome, and then they would do the treatments after that. But they found it was so harmful for these mice to not have any, they literally couldn't function when they
had like neurodevelopmental, like disabled, basically, like they didn't develop properly.
Yep.
Which
so
is
I
crazy.
think that speaks very highly of how important the microbiome is to our, just our basic level of functioning. And so in terms of current literature, tons and tons of data in mice. And a lot of that we're seeing isn't necessarily translating as well to humans in terms of practical, like clinical application. The most, I feel like, profound things that we've seen are these fecal microbiota transplants, where we're really seeing that in humans who are really suffering with different chronic diseases, that we are able to see these profound shifts in disease outcomes and states when we do fecal microbiota transplants. And there's a number of different issues with that, of course, because, we can't, if we have a single donor and donors' microbiome is changing and evolving all the time. So we don't know if we're going to get the same population of gut bacteria from that single donor. It's also kind of like an organ transplant or a stem cell treatment where you need to make sure that it's safe and that there are of course risks involved, right? Where you could be delivering pathogens to the recipient from the donor as well as good bacteria. So you have to be able to kind of screen the entire population. So there's we've seen these positive outcomes, but there's still a lot of things that need to be fleshed out in terms of risks and consistency before a fecal microbiota transplant would become a regular thing. But we are seeing that's kind of probably the most profound example of research that has been done to date to really demonstrate how profound some changes in the microbiome can be for helping human health. So that's kind of on the clinical standpoint. In terms of like history and like where this all started, we can go all the way back to the Human Genome Project. So we have, you may have heard of the Human Genome Project or the Human Microbiome Project. So two different projects. The Human Genome Project is looking at our human cells and all of the genes in our human cells. I think this was back in 2003. And actually, we have a colleague who worked on this project, Dr. Susan Trapp. So shout out to her. She was working with Craig Venter, I believe, on this project. And so they successfully sequenced, I think, 92% or so of the human genome. And what that did was look at this blueprint of human DNA. And what they found actually was that our human DNA was highly dependent on microbial DNA and all of these other biomes in our body, right? So not just the human genome, but the microbiota genes as well. And so there was kind of this discovery that there's up to 100 times more genes in these other species that live in our bodies versus our own cell DNA. And so that was kind of the major discovery there, was that there's actually this huge interdependency on the microbiome. So then we had the Human Microbiome Project, which was launched in 2007 by the NIH, and the Human Microbiome Project. I used to think this was where they looked at the gut microbiome, but they actually looked at the microbiome from several different organ systems. So they had the skin, the lungs, I can't remember all 5, but they looked at these different, the skin, mouth, GI tract, the urogenital tract, and the nose. So they looked at 300 healthy adult volunteers, and they used metagenomic sequencing to identify and characterize these microbial communities. And the key finding from the Microbiome Project was that there is a core microbiome that exists in healthy adult humans. So they kind of found these. So of course we have genus and species for bacteria. They found that there was this core population of different bacteria that existed, but that it varied significantly between people. And of course, this study didn't look at everyone from infants to the elderly, different sexes, different, geography, right? There's so many different things that come into play, different diets, all that sort of thing. But we did, find this core microbiome, at least in healthy adult volunteers. They also focus primarily on bacteria. And like Morgan, you mentioned earlier on that the microbiome actually has a virome and a mycobiome for fungi and archaea, right? There's these other biomes that exist as well. So that study primarily looked at the bacteria and didn't do a deep dive into these other regions, which were probably a little bit more complex to study, I would imagine. And even the bacteria there are certain strains that have been proven more difficult to study, specifically the anaerobes. So we have bacteria we have aerobic bacteria that utilize oxygen, and then we have anaerobic bacteria, which if they are exposed to a single molecule of oxygen, they can't grow and they die, right? So we have these anaerobes, we have many anaerobes in our gut. And so it can be difficult to study, particularly the anaerobic species. And then we also have what are called facultative anaerobes. And that basically means that they're able to use oxygen or not use oxygen. So they can kind of go between those two states. And so some of the issues that have come up in these projects with understanding the microbiome has been, and specifically the gut microbiome, I'll say, is that we can't always study some of these species and without killing them and get like a perfect idea, right? And it's always evolving. So these studies, if you sample one time from a patient you're not really, it's about the evolution and how it's changing, right? So there is this core microbiome, but then everything we do, eat, what we eat, our exercise levels, antibiotics, processed foods, and all these things are impacting stress, are all impacting our microbiomes. And so what we really need to do is continue to sample over the lifespan and over different situations and different contexts to be able to fully understand it. But that's kind of, I would say, the summary of where we are today in terms of we've sequenced, most of the human genome, and we've sequenced, I think I've heard that we've sequenced all of the gut microbiome. But again, if you have these different populations and different circumstances, have we truly sequenced all of it? Or is that, just the bacteria? And even in that, like, which species are different and which populations, I think it's definitely always evolving. It's a field that's continually growing, and we're continuing to do more research. So I would say that's where we're at.
Yeah. It's crazy too to think, the Human Microbiome Project was done in 2007,
Yeah.
right? And there has been such an advancement in like our technology and our ability to study microbes since then, that really that was, while we learned so much from it, that was, like the tip of the iceberg in what really is happening in the human microbiome. So cool thing to be looking forward to in both medical and science, right? Is like just what could we still have to learn about all of this?
I literally just read this morning, I heard on, I think it was like Medscape or something, they were talking about coffee, and they said there's a certain population of bacteria that are found to be four times more prevalent in coffee drinkers. And Nick said to me, why does that matter? I said, I don't think they've gotten there yet. They just found that there's more of a certain type of bacteria. I was like, they're probably good at metabolizing caffeine or something, but there's got to be something.
Ooh,
Would that be great, right? I like to think, I love coffee, so
yeah, metabolize all of the like bad, quote, bad parts of coffee that everyone says are bad for you, and like, yeah,
exactly.
You just don't have it.
Just straight antioxidants.
Your little microbes
now.
are doing their job.
Polyphenols, which we'll get to, but.
Yes.
I was like, I
it.
don't think they flushed that out yet fully, but it is interesting that they have found a correlation with different bacteria.
I feel like, so we talk about in research a lot, like correlation versus causation, right? But I feel like when you talk about the microbiome, it is no more prevalent than like, okay, is it a correlation or is it a causation? And it's just like, there's so much that the microbiome does that we just like don't really understand yet. So
yeah. The function
so cool.
piece is definitely, so yeah, there's and even, I think I skipped over this part too, but you have the central dogma. If we go back to, biochemistry 101, you've got DNA, which turns into RNA, which turns into protein, and the genes don't always line up with the proteins and what's actually happening in the body. And so there's a lot, and even that, like the proteins can get modified. And we have things like methylation. So, and this is the whole conversation about everyone thinks, oh, if my mom or dad had a certain disease and it's genetic, I'm going to get it. Not necessarily, because of epigenetics and methylation. And so the same thing is true with these bacteria. They have genes. And when we're looking at the genetics piece, we're not necessarily seeing, these end stage proteins and the functional pieces of what's going on. And we haven't explored that. I haven't read anything yet about how, the genetics of these bacteria are getting translated to the functional aspect. So can you imagine, like, we're still trying to figure that out in humans, how do we adjust this genes to manifestation of disease and symptoms pathway? So it's, and when you and I work in the research lab, we're always doing both, right? We're always looking at the PCR or the genetics piece, and then we're also looking at or RNA-seq, and then we're also looking at the protein side of things with things like Western blots and ELISAs and the end stage piece of the protein. And sometimes those things line up and sometimes they don't. And it's interesting, or you have like a huge amount of genes, but then what actually shows up in the protein is a lot smaller. So by the time it gets there, because only parts of it are active, and so are like translated all the way. So I think there's a lot to be explored there too, in terms of like, which thing are you looking at? And I know in the research lab too, you're only able to grow certain things, like culture them in a petri dish, and so, or cell culture. So like if you're doing the genetics, you're going to see different results than if you try to culture something. And a lot of the labs that I use clinically will do both. So they'll look at both the genetics, they'll look at under the microscope, and then they'll also culture and see which things grow over time. And certain things are just, like we've talked about, notoriously hard to culture because of their anaerobes and can't be exposed to oxygen and that sort of thing. Or they're just difficult to grow. And some certain bacteria, we found ways to grow them easily and optimally, like lactobacillus and bifidobacterium. And that's part of the reason that those are so prevalent in probiotic samples that you'll see, is because they are easy to grow. And so there's all these kind of, just state of microbiome situation.
Totally. It's just not a straight line, right? That's
it's complex, right? I see different numbers all the time. So I don't even like throwing out numbers. Like we have 100 times more genetics and we have 2,000 species, and we have like all these numbers that, this many trillions of cells or bacteria, like we know it's in the trillions probably. And we know like there's certain things that we feel pretty confident about. And there's other things that every day it seems like there's more evidence. And so I have a hard time even like putting numbers out there because like we're still learning about it all the time. Do you want to kind of talk about the establishment of the microbiome, kind of so that's where we are with research, but like as humans, we obviously get our microbiome at birth, right? Or, growing in the womb and then through delivery. So can you kind of talk about the lifespan of the microbiome in humans?
Yeah. So the microbiome in general, so we have early life, so infancy, right? And this is where we have this rapid microbiome change in the first year of life. So we go from like, being in utero, we get exposed, and we'll talk about this in a second, but we get exposed to all of this microbiome when we are born, whether we're born vaginally or versus cesarean. Either way, we're being exposed, but there's like some interesting diversion in what we're exposed to depending on how we're born. But just as everything in the first couple years of life, we're growing rapidly, right? So our microbiome is also changing rapidly as we go from either breast or formula milk to solid foods, our environment, like what medicines or antibiotics we're exposed to. We really hit peak diversity in adolescence and young adulthood. So I heard this described a podcast with Dr. Peter Attia, where he was saying the time in life when you can literally consume anything, you can not sleep, you can eat all the foods or drink alcohol or do anything you want and not be really affected the next day. That's kind of evidence that you're at your peak microbiome. Like they are able to like handle this kind of onslaught of abuse that we all put ourselves through in our like teens and maybe even in our early 20s, right? So that's kind of our peak highest diversity. That high diversity is really correlated with the better health outcomes. We're really disease resistant. Like it takes a lot more for us to get sick at that time. And then after that peak in that kind of late teens, early 20s, we really start to just kind of drop off for the rest. It's the decline of aging, right? So we have reduced microbial diversity as well as just, I think, colonies as well. And it's associated with increased inflammation. We have chronic diseases, that kind of the frailty of the body as we grow older. And there's several factors that contribute to the development, but let me rewind to birth, right? Let's start there and kind of go into that initial seeding. So now we have like a view of what our microbiome looks like over the lifespan, but how does it really, really get started? And when we are born, we pass through typically the vaginal canal, right? And we are exposed to fecal bacteria. And there's a couple things here where I know Dr. Keeley and I were kind of like, huh? Like, wait, what? And, you hear like, oh, you're exposed to like vaginal flora and yada, yada. But like, what does that actually mean? And so to kind of like rewind a little bit, there's this like big microbiome change that happens during pregnancy in the mother, right? So they go from having this really diverse vaginal microflora to actually during pregnancy, it like reduces down. You pretty much only find lactobacillus in the vaginal canal, which I think is so interesting. And they've associated a change in vaginal microbiome to things like like cervical insufficiency, so difficulty carrying pregnancies. They've also related it to bacterial vaginosis can be a change in the microbiome, and that's related to preterm labor, all sorts of things that this little microbiome in the vaginal canal really affect, which is bananas to me. And then the cool thing is, as hormones change throughout pregnancy, so as we're approaching like full term, right, and they don't really get the mechanism of like why this happens, but some of it's related to an increase in estrogen. It causes gut microflora to actually like migrate to the vaginal canal. So now the vaginal canal right before birth is populated with a lot more micro like colonies, like different colonies, right? So it could include things like E. coli, Enterococcus species, Bacteroides species. So we go from like just that like pretty strict lactobacillus during pregnancy to all this like diversity. And as the infant passes through, that's where they really like get exposed to all of those things. Now, during C-sections, right? So we're still exposed to bacteria, but they found that what happens is a lot of C-section babies are really more colonized by the microflora that you would find on skin versus like the gut and the vaginal microbiome, right? So, and they're also going to be colonized with more of like environmental bacteria because they're kind of exposed to the environment before they're really exposed to like the flora of a body, right? It is associated, they say lower microbial diversity is seen in C-section babies, which can lead to a higher risk of allergies, asthma, obesity, and autoimmune disease. And I'm trying to think, did I cover all
Yeah,
that?
I think you covered. It's just wild to me that like I see a lot of crazy stuff associated with like estrogen shifts in the body in adult females, not necessarily just pregnant females, but I think it's amazing, like these little things that we're kind of exploring, learning that can I mean, pregnancy, we know changes everything, right? But even something as subtle as maybe this
Yeah.
hormonal shift leading up to birth that could potentially change the vaginal flora that then translates to the baby. Like it's just so cool. And I remember here, like when we first learned about delivery and learning that there was X number of processes in the infant that had to shift immediately after birth for them to be able to breathe on their own, for the heart to, start pumping, like just all of these details that have to happen physiologically. Yeah. I just think about that too with this microbiome shift. It makes so much sense that it's all happening for a purpose. And yeah, you did a great job covering like pregnancy and then the birth and then even into breastfeeding with the infant, right? There's more that goes on with breastfeeding that also helps to like continue establishing the microbiome, right? So once the baby's delivered, we have either C-section or vaginal delivery are kind of the two methods, and those establish a different initial microbiome. And then we get to either breastfeeding or formula feeding. And there's a couple things associated with that. So with breastfeeding, and I just want to say too, though, that this is in no way meant to like, mom shame or add any sense of like guilt. Because I feel like it can be a sensitive topic to discuss these things. And I think part of the exciting time we're in is that we do have potential interventions for these things as well, right? And they've explored adding like bathing the baby in the vaginal fluids after birth if they're C-section delivered. And there's been some controversy. Well, what about pathogens, right? Or like, what about, can we do that safely and effectively, right?
Yeah.
Which I think would be really cool if we could figure that out. But they're trying to come up with solutions to these things because we know they're a reality. But I just kind of wanted to mention that it's a part of the whole process, right? To figure out what's best for you and your baby. And so when we get to breastfeeding, if an infant is breastfed, we have different types of sugars that we consume. So we have like monosaccharides, disaccharides, and something called oligosaccharides, which is a type of sugar. And in breast milk, we have human milk oligosaccharides. And these are actually sugars that don't get consumed by humans. So the infant won't process these, but the bacteria in the infant will process these HMOs, we can call them for short. So the HMOs get consumed by the infant in breast milk, and then they are processed by largely bifidobacterium, a species in the gut. And these bifidobacteria, they actually produce things called short-chain fatty acids. So that's an end-stage product of bacteria. Those short-chain fatty acids, namely acetate and lactate, those can feed other sources of bacteria in the infant gut. And that's where we get more microbial diversity. So initially, breastfeeding, we have the HMOs, the bifidobacteria digest it, produce short chain fatty acids, and those feed other gut microbes. And then we see this increase in microbial diversity, largely due to that initial bifidobacterium population. And so this cross feeding just supports the growth of additional species and increases that diversity. And then in formula fed infants, we tend to see actually a greater diversity of bacteria, which is interesting, but we tend to see more pathogenic bacteria. So, or fewer beneficial species, I should say, not necessarily pathogenic, but we don't see as many of the beneficial ones, such as the bifidobacterium. And that's kind of the main thing between that we know between breastfeeding and formula. And of course, this is also an evolving field. So these are just like kind of the general overview that we see from research so far. Another thing about breastfeeding is that it can enhance the immune system development. So when we have those HMOs, this helps to reduce inflammation in the gut, and it also supports the maturing of, we've talked about this in previous episodes, but the GALT tissue, or the gut-associated lymphoid tissue. And that's a big piece of the immune system that develops in the gut. And if we know like 80% of, we've mentioned this before, 80% of our immune system starts in our gut. And so when those bacterium are enhancing that GALT tissue, we're really helping to establish a strong immune system in the gut. And that helps us protect against those pathogenic species as well when we have a stronger immune system. Oh, there's also one other cool feature of the HMOs. So they can actually supposedly mimic binding sites of the gut epithelial cells, which can prevent certain pathogens like E. coli or C. diff from attaching and colonizing. So these HMOs from breast milk, basically they're competing for pathogens. So if you have this HMO in the gut and you have a gut cell and that pathogen binds to the HMO, it's not going to bind to the gut cell. So some will still bind to your gut cells, but if some are binding to the HMOs, then you have less pathogens that can bind to the gut and essentially establish colonies along the gut wall. So they're kind of this, just doing all these different things, right? So we're feeding the good bacteria, producing those short chain fatty acids. We're helping to build the immune system, reduce inflammation, and we're competing for pathogens, right? Which is why maybe we see that slight imbalance in microbial species with formula feeding. So I just think it's so cool, right? This is just what we know so far. I'm sure it's so much more complex than that, but that's kind of, and neither of us are an expert in this topic, but I think, just to see the little that we're starting to figure out about these things, like everyone kind of knows, right? Like, oh, it's optimal to breastfeed and, it's optimal to have a vaginal birth, right? But what's the why? How are all these things happening? So I think it's super cool.
Yeah, absolutely. And I think to kind of like one thing that we didn't touch on is what we're really seeing is that the microbiome is actually really flexible. It's really changeable and it can like change really quickly. So just as like a note on the like, well, if you were formula fed or if you have an infant that's formula fed, it's like, well, there's ways to introduce those like good, well, I hate the word good, right? Like to those more ideal bacteria. And like, it's so flexible. It's crazy just how much you can change your microbiome by changing your diet or changing your environment or, getting out and getting in the dirt and doing some gardening and being exposed to other microbes. There's a lot of opportunity there to change and establish a different microbiome.
I'm really curious to see future studies on like damage to the microbiome and how that impacts the ability to change your microbiome, because I have heard about like damage to the crypts in the small intestine where they can actually like get stuck and form like permanent pockets and biofilms, and how like damage to the enteric nervous system, right? If you're not able to like continue moving pathogens out, right? Like how does that impact? I mean, like biofilms in general would be a whole topic for discussion of how are biofilms in the gut like persisting versus other, and how do they continue to feed it, right? I just think there's so much there where we're like, yes, it's versatile. And it seems that some people have like stubborn, persistent gut issues their whole life. And some people have how does the damage impact it? And like, can that be healed? Or like, are there things we don't know yet? And like, would an FMT make a difference? Or like, these crazy high dose probiotics? Or is it, are there some things that like aren't reparable? Like I'm so curious to see more data evolve on those things.
Yeah. And long term, right? Like, what does that look like? I mean, we know our cells turn over like over time. Is it a long-term impact? I don't know.
Yeah.
Right. So interesting. Cool. Well, let's touch a little bit on like the actual species. Like, what are all these different microbes doing for us?
Yeah, so when they establish kind of the core microbiome, there do seem to be these like really predominant, I guess it would be the genus, right? So we have the genus and the individual species, and so there's kind of some key groups that are in the healthy human gut. So we have Akkermansia, we have Lactobacillus, we have Bifidobacterium, Firmicutes, and Bacteroides are kind of 5 of the core genus or genii. I would say that they would find in the gut.
Yeah.
And Akkermansia that's kind of a hot topic right now. People are all about Akkermansia because they're finding that it does seem to be about 10% of the gut microbiome in healthy populations, and they're finding a lot of diseases associated with lower amounts or no Akkermansia species. And so this has been a really hot topic for cardiometabolic health and weight and other chronic conditions. These Akkermansia feed on the mucin layer in the small intestine. And so they actually help. That sounds like a negative thing initially, but it actually helps with the cell turnover and the regeneration of the gut lining integrity in the small intestine where we have that mucus layer. And then Lactobacillus produces lactic acid, which is associated with gut health and immune balance. Bifidobacterium, we talked about it being predominant in breastfed infants and helping with digestion and immune function. And then the Firmicutes and Bacteroides are regulating energy metabolism and nutrient absorption predominantly. So again, lots more to explore there in terms of function, but that's just kind of a high level overview on the core species. You will see, on that note, you'll see Lactobacillus and Bifidobacterium species in just about every probiotic that you find, and a lot of them will be like solely those groups with different species. And the main reason for that is because these were some of the earliest discovered genus, right? Genii. I really need to go look that up. Do you know which it is? Is it genii? Oh, this is bad.
No. Let's do
a quick search
here.
so I don't keep saying it wrong. But the Lactobacillus and Bifidobacterium they're also easy to grow. So these are easy to culture and produce a lot of because they're not anaerobic species. And so we can get a lot of them, and they're cheaper to produce because of that. It takes a lot more to produce something like Akkermansia, which is an anaerobe and can't be exposed to oxygen. So that's why you'll see predominantly those species. It doesn't necessarily mean that's what you want to be taking. So do you have an answer?
Genera.
That's it. It's been a little while. I was like, genii does not sound right. Genera, maybe? Genera. I clearly don't say that often.
Yeah, wherever.
It's funny.
Genera. Yep. It even gave me like a pronunciation. So Jen dash ER dash UH. So genera.
I think it's genera. I don't know. Someone correct us if we're wrong. Okay. Moving on. So we have the core microbiome, and these are positive species, right? Okay. So when you at least we learned in gastroenterology and naturopathic school that you have kind of the good bacteria, the bad bacteria, and then you have the opportunistic bacteria or the commensal bacteria, which are a part of your normal flora. But if things are out of balance, you take an antibiotic or heavily processed food diet, sedentary, all these things that kind of reduce your positive populations of bacteria, then you have an opportunity for these bacteria that are kind of kept in check to then overgrow and become bad bacteria. But the better conversation is the balance and the diversity of your bacteria. So do you have them in the first place, and in what prevalence or abundance? And so a lot of the stool testing, like if you do a lot of functional medicine stool testing, you'll see them labeled this way, like, positive, negative, and opportunistic, right? They're usually in like a green, red, and yellow, right? So you're kind of like, these are the bad guys, these are the good guys, and we want these in abundance, and we want these guys in check. And so talk about some of those opportunistic ones where we definitely want to keep them in check, and, what happens if they overgrow, that sort of thing, or what are some of the main ones that tend to overgrow?
So as far as so these are bacteria that we do have in the gut. So we have Clostridium difficile, and then E. coli. Everyone's heard of E. coli, right? And then we have Klebsiella. So these are all in the gut, but they're usually in really small amounts. So they really only become problematic if, like she said, we take an antibiotic, or we have some sort of health crisis, or have a highly processed diet, situations like that. Now, there's a set of species that are not colonizing the gut, but we can get them. We usually get them from an outside source. That would include Salmonella and Shigella, and then a certain toxin-producing form of the Clostridium difficile.
Or C. diff is like the
C.
shorthand
diff,
that everybody
yeah.
wants
So
to
everyone's
probably recognize, right?
Yeah, C. diff is like the thing you get in the hospital, right? That everyone's terrified of. And I was listening to a researcher earlier this week, and she was saying that C. diff isn't like actually super studied because she thinks like everyone's scared of it. So there's like things that they haven't, not that it's not studied, but it not as studied as it could be, right? So yeah, so we have opportunistic, so and then toxic, right? Sorry, to circle back. So we've got these three main categories: the bacteria that colonize, that do all the things in our body, digestion, immune, metabolism, all of that. We have the opportunistic pathogens, which do colonize our gut, but in small amounts and can become problematic given certain conditions. And then the toxic species, which we can pick up from the outside world and can lead to sickness, illness, disease, that kind of thing.
And I know, oh, sorry.
So
I don't want to interrupt you.
Yeah. No, you go.
I was just going to add too that like people, that's kind of up for debate, right? And I think as we learn more about the functions, we'll have more clarity in like, do Salmonella and Shigella have any positive functions, right? Because you don't find them in most of the normal healthy population, but they do seem to exist in some people and not cause harm. But are they doing something beneficial? Or in that particular host, are they doing something beneficial? I think that's where the function piece comes into, of like, how do we categorize these as, do you never want them or do you want them, right? I know there's certain like parasites too, like Blastocystis hominis that like a ton of people have, but they're still trying to figure out, is it actually causing symptoms? Like, is it a symptom producer or only in certain people? Or is it, is it beneficial or harmful or neither? And I think those are some of the answers that we're still looking for to determine how to categorize these things. But at the moment, that's kind of like the lay of the land in terms of identifying these species, right? We know those ones produce toxins that directly attack the gut and can cause, bloody diarrhea. Like that is probably, in my opinion, I'm like, it's typically a negative
Right.
bacterium. But if it's, but maybe in some people it serves a purpose and we just don't know that yet in tiny amounts. So I think that's where we have room for exploration. So do you want to talk about diet and kind of like, okay, so the next thing I would say with the microbiome, like people have finally kind of like come to understand like what a probiotic is. We also now have prebiotics and we have postbiotics. So can you kind of delineate really simply what is a prebiotic, what is a probiotic, and what is a postbiotic?
Yeah, totally. There's a lot of confusion around this, right? And I feel like even myself, I have to kind of like look it up every time to make sure I'm saying it correctly. So prebiotics are non-digestible fibers, and these are what feed the microbes. So we commonly see this. So inulin, which is
the most common one,
a derivative of chicory root. Yeah, pretty common. Pectin from apples or fruits. So fruit pectin. And then some sort of resistant starch, which we can get in something like a banana. So fiber is really a non-digestible. So, and when we say non-digestible, it means literally like humans don't digest this fiber, which I think is so interesting that it's really so important for our bacteria, but not really important for us, but important for us because it's important for our bacteria, right? So that's prebiotic. Probiotics are actual live bacteria, or like freeze dried, or what do they call it? Like live attenuated, bacteria. So that's where you actually get like a little capsule, and that little powder in there is the bacteria. So we can get this naturally. We get this through fermented foods, right? So we could have it in yogurt. I think yogurt does, like natural yogurt has a certain level, but the yogurt we buy in the grocery store definitely has added probiotics, and probably a lot of these do, to be honest. We see it in kimchi, we see it in sauerkraut, so pickled foods. Common species there, of course, lactobacillus and bifidobacterium, like we said before. So then we have postbiotics. So postbiotics are actually the metabolic byproducts of probiotics, of those bacteria. And those include things such as short chain fatty acids. An example of a short chain fatty acid is butyrate, and those help reduce inflammation and maintain gut barrier integrity. Another thing that I learned this week while researching for this was that butyrate is one of the short chain fatty acids that helps in GLP-1 release in the gut. So GLP-1, that's like a big hot topic right now with certain medications on the market. And so this can help with appetite, satiety, fullness, and glucose regulation in the blood. So interesting, right? So that's postbiotics. Then we have a term, just kind of like I guess maybe an industry term, we could say it's synbiotics. So this is really just a combination, and we would see this in our like nutraceutical, like our on the shelf prebiotic, and it can be a combination of either prebiotics, probiotics, postbiotics, any combination of the above. So that those have been put out by nutraceutical companies to kind of like create this like comprehensive product where you get that prebiotic and the postbiotic. So you're essentially providing the food for the bacteria as you introduce the bacteria to your gut, and so you can feed them and colonize at the same time. Kind of that thing. And maybe you can talk a little bit more about like the potency and like what does all that mean? Because when we think about the actual on the shelf, like you see all these numbers, and I know I've stood in front of a prebiotic, postbiotic aisle and been like, I don't know what any of this means. So maybe you can help clarify.
I can't tell you the number of times that someone has sent me a picture and said, is this a good probiotic? And it has like 5 million bacteria in it. And if you think about the trillions of bacteria in your gut, is that going to make a dent? Probably not. At least I haven't seen it make a dent. So in general, yeah, we have this wide range from, I would say, like in the low millions up to hundreds of billions. Like I've seen them up to 500 billion colony forming units, or CFUs, which is kind of an arbitrary term. Theoretically, it's the number of probiotics. But yeah, anywhere from 5 million to 500 billion. And there's different uses and different purposes, right? And we don't have a lot of treatment data on, which potencies to use. We have some studies of patients taking antibiotics and taking a certain dose of probiotics during antibiotic treatment or after antibiotic treatment, and the jury's kind of still out on that one, because essentially it's a space occupying issue, right? We have gut, and if we take an antibiotic or eat certain foods, sedentary lifestyle, whatever, all those things are going to knock out certain populations, and you have space. And then that space can be filled by either diversity and good species, or it can be filled by harmful species or opportunistic species overgrowth. And so when we think about knocking out those bad guys or filling up that space, right, we want to make sure we have enough. And so in general, most people taking, a daily probiotic or taking it for gut health or whatever are probably going to be around the 25 billion range is kind of a typical standard probiotic you might see out there in the market in the mid-range. And then if you're taking typically those ones up in the higher levels are for certain conditions, or I've used them personally when traveling and immediately kicked a GI bug when traveling in foreign countries. I'll often take that and Saccharomyces boulardii for acute traveler's diarrhea, that sort of thing, and that combo. And Saccharomyces has been very well studied for diarrhea and that sort of thing too. So that's a whole nother conversation. But yeah, not medical advice, just what I do personally, and kind of state of the affairs. But yeah, very wide range. And I guess the other question is, do you want to be taking, and these are probiotics specifically, these are the live strains of those bacteria. And we have also like stability, like refrigeration, enteric coated, like is it all getting there? And how much is in the probiotic at the time of manufacture versus will it extend the entire lifespan until the expiration date? And there is no guidance or regulation around that. So you could be seeing, 5 million when it was created, before it was freeze dried and packaged, and it might be nothing in that probiotic, or it could be dead. And if you're working with things like Akkermansia that are anaerobic, those are also going to be a lot more expensive because it's just more expensive to produce that kind of probiotic. But the other question would be, do you want to be taking prebiotics and postbiotics or just probiotics, right? And we're still figuring that out, right? These symbiotics are pretty new. And I will say I've seen like crazy things happen when you ramp up the prebiotics too fast. And so that's definitely something where, think fiber, right? If you eat too many beans and you're not used to having a high fiber diet, you're going to feel it, right? And it's the same thing if you try to just, add all of these prebiotics into your diet immediately. Like something like inulin can really set people off, especially if you have something like SIBO, small intestinal bacterial overgrowth, or like some of these other conditions. It's really going to take a toll on your gut. So go low and slow with the prebiotics if that's something that you want to experiment with. But they do really augment the effectiveness of the probiotics. And you'll see it in things like yogurt too. You'll see something like, inulin or certain types of resistant starch in the yogurt along with the probiotics. And so it's kind of built into some of these things already in addition to, the symbiotics, but definitely new and exciting fields.
That's a great segue right into fiber. And I know you're really excited about fiber right now. So talk to us about fiber.
I love fiber. And really the main thing about, we're going to do a whole episode on fiber, I think, because I am just like nerding out about it. And I've been trying to get all my patients to eat more fiber because it truly is like the source of diversity for your gut microbiomes. You should be trying to, basically Americans get, I think the latest statistic I saw was like less than 5 grams of fiber on average per day. And the recommendation for men and women is like 25 grams per day. Men even more, like 30 to 38 grams per day. It depends on which authority you're looking at for the guidelines. But in general, most of us adults need 25 grams or more of fiber per day. And our ancestors, there's some evidence that they ate over 100 grams of fiber per day. So like our genetics and our systems are really set up to be fiber consumers. And I know there's people on carnivore diets that have like perfect metabolic health and have like adapted that way too. And I'm really curious to see future research on what microbiomes look like in carnivores and, ketogenic diets and some of these diets that tend to be a little bit lower on, the complex carbs even and fiber sources. But in general, our gut loves fiber. And so it loves fiber and it loves polyphenols, which are also found in fruits and vegetables and fibrous foods. Polyphenols are the antioxidants that plants produce to defend themselves from attack. So they're essentially plants, right? They can't run away if there's an invader. And so they have to produce these metabolites to defend themselves. And one of them is polyphenols. And so fruits and veggies have really high amounts of these polyphenols that are also consumed by your gut bacteria, not necessarily by you. There's been complaints that polyphenols aren't super bioavailable, but it's because your gut is really using them. And so it's your gut microbiome that wants the polyphenols and then, maybe doing some additional work in the body. So we don't care so much necessarily if it's getting into the bloodstream and it's super bioavailable, if it's mainly acting on the gut flora. And so that's kind of the current thought is, fiber, polyphenols. In terms of the other thing about fiber is you want to it's the diversity too, right? So it's the grams and it's the diversity. So we want, that 25 gram plus, which it may seem hard to do if you're eating a standard American diet, but like a cup of raspberries has 8 grams, right? So, there's, things like lentils and oats and beans, like there's ways to get a significant amount of fiber in your diet. And there's so many other benefits. So we'll go into that later, but it's the amount and it's the diversity. So a good goal is to shoot for 30 different types of fiber per week, which also sounds insane, but you can count herbs. So I think I was saying to you earlier, like if you think of a taco, right? Like you've got 10 different sources of fiber in a taco. So you can still, if you put some pico de gallo on it and you have, tomatoes, onions, jalapenos, cilantro, you can count the herbs, right? There's cayenne, there's, if you put avocado or guacamole, cumin, you've got
cilantro,
lemon, lime, like there's other things, right? So you can easily get to 10 eating a taco. So I think, it doesn't have to be as challenging, right? And salads are really good too. Lots of diversity. You can get 3 different types of leafy greens and that sort of thing. So a lot of my clients ask, like, can I just take like psyllium husk fiber? And like, you can, but even those, like they might only have 3 or 4 grams of fiber in a serving and you're going to have to go low and slow. And obviously there's the polyphenols that come with the whole foods and things like that. So yeah, we'll just have to do a whole episode on fiber and deep dive on that because I think there's a lot there in terms of metabolic health and all kinds of things. So
we have
key, do we want to just kind of wrap up with any last thoughts on the microbiome? Like what other things do we care about with the microbiome? Just at a high level.
I mean, they impact everything, right? So like a big thing for us, right? We talk a lot about mental health on our podcast here, and the microbiome in the gut is really affecting that gut brain axis. So we've talked about that, like the enteric nervous system, the vagus nerve, the brain, and like neurotransmitters. I mean, the depth of this is vast, right? And I think that's kind of like where we're kind of like sweeping back around to, right? The impact that our microbiome can have on our mental health as well as our like overall wellness, and how much the microbiome is communicating with the brain via the vagus nerve, via neurotransmitter production, right? How much neurotransmitters are created in the gut, right? So thinking about all the different ways that imbalanced microbiome can affect and like increase anxiety, lead to depression, cause cognitive decline, and things like that. I think that is kind of where you and I are headed with this, because just to kind of like refocus, but there's so many rabbit holes, right? We could go down with this, because it is what we're learning more and more, is that it's so impactful for so many facets of health.
Yeah,
right?
I admittedly would sometimes get frustrated when I would go to doctors early on and they would all be like, well, I'll have to go back to the gut. It all starts in the gut. I'm like, really? We also have like hormones and, the nervous system, all these other things, right? And it truly is the central hub of so many things. And especially in like our culture and our lifestyle with, the food we consume and just everything that we're exposed to, and the stress, this is all impacting our microbiome. And we're just like, it truly is the center. And I think if you have a lot of things going on in the body, it's definitely the first thing you need to look at in all situations is gut and gut health, right? And we can talk about, I think, the gut testing, and for future episodes, kind of like what does functional gut testing look like and what information can we find out? And there's some of these new companies that are doing like personalized gut testing, and they've done studies on autism, also showing like if we give you a customized probiotic, we can improve gut health and, autism or mental health symptoms associated with autism as well. And so we're seeing like all of these kind of progressions in this field. And so I think we can, dive into some of those in future episodes too. And also some of the harmful things, right? We didn't really touch on like, we kind of touched on what helps the microbiome and establishing it. We didn't necessarily touch on like the things that may be impacting our microbiome that we should also be careful with, like artificial sweeteners, high glycemic foods, alcohol. Like we haven't explored how a lot of these things could be impacting our microbiome and our nervous system via the microbiome. And so I think that's kind of where we can take some of our future episodes, because it really is super central. So I know we didn't focus a lot on that nervous system component, but I think that it's the immune system, right? It's hormones, it's neurotransmitters, it's inflammation and systemic inflammation. It's all of the things, right? And so it's super important to get that sorted out, right?
Yeah, totally. I mean, this whole thing, right? Like I guess like the overview to zoom out for today is like microbiome is complex and it's super important, and we can support it by eating a variety of foods, a variety of fibers. What else? How can else we support it? Limiting
high fiber. So
processed
25 grams plus per day, limit ultra processed foods, have an active lifestyle, hydration, like potentially looking at your diversity, looking at your microbiome, seeing how you can improve, that sort of thing. And then of course, like, breastfeeding, we kind of talked a lot about that, about like the establishment at birth and not taking, or like being grateful in your teens and 20s that you're having optimal diversity. And if you're on the decline, maybe working a little extra hard to increase that, fiber, polyphenols, diversity, that sort of thing. So
completely.
Are we gonna skip our question for today?
Amazing.
Time, I think maybe we could do 2 questions next time.
Or
just a really good one
next time.
I'll be like,
I love that. Yeah, let's do that. All right, so that's a wrap for us today on the microbiome. We'll be back next time with a deeper dive into more juiciness about the microbiome. I don't even know how to explain that or how to tease that, because it's like sometimes we don't know till we know. And here we go.
A lot of fun.
So thanks for tuning in.
We're a lot of fun.
Thanks, everyone. I know, right? By the seat of our pants here. This is, we just do what we are interested in. This is
what
the
we have
things.
time for.
Anyway,
it's going to be.
Exactly.
Thanks for hanging with us.
All right. Thanks, friends. Yep. See you next time.